All right, hello everybody. Today we're going to talk about experimental design. How do we do an experiment? Because this is a science class, it is really important for us to know how we do experiments. But before we go into how to do experiments, I want to show you how you're going to set up your lab notebook for when we take notes in this class. So the first thing you're going to do anytime we take notes, and you should do this now because you're going to take notes here, is take your notebook and draw a line right down the middle of the notebook. So I will show you what that looks like. You draw a line right down the middle of the notebook, just like this. Then, every time we take notes, we'll start off by showing you a series of questions. So when you get those questions, you'll write them down on the left side of the notebook. So you'll write them down right here. Now, notice how in this example, there's space left between each question. This is because on the right side of the notebook, you'll write the notes that correspond with each question. This means that your notebook will be really easily laid out. If you need to find information, you can search for it by scanning the questions on the left, and then you can find the information that you're looking for on the right once you found the question. So for this lesson, we are going to be answering 8 questions. We're going to answer what is an experiment, what is your question, what is a hypothesis, what is a variable, what is an independent variable, what is a dependent variable, and what is a controlled variable, and also how do I collect data. So in your notebook, you'll write down each of those questions in the left-hand column. Now, it might take a couple of pages for you to get all of these written out, especially if you are leaving the appropriate space between each question. You should know about how long it takes you to write things. If you have really big handwriting, you might want to leave more space than if you have very small handwriting. And you'll start to get a feel for this the more we take notes in this class. But you should set it up something like this. I wrote kind of big. You can obviously write smaller. But you see how I wrote the questions on the left-hand side. My right-hand side is currently blank. And I left some space here so that I'll have plenty of space to write the notes in. So if you need more time to write these questions down, you can pause the video, but I'm going to move on. So the first question we're going to answer is, what is an experiment? And an experiment is a scientific test that we use to learn about the world. So what you're going to do in your notes, right next to where you wrote down the question, what is an experiment? You'll write down the explanation that we provided. So I'm going to write an experiment is a scientific test. So you can see how in my notes now, I have the question here and the answer here. This is how all of our notes in science are going to be laid out. Now, one other thing that I recommend that you do in your notes is to draw little pictures. So you should have some space between your questions on the left side. And I like to use that to draw in little images to help me remember the material better. So you can see under what is an experiment, I've drawn an ugly little beaker here. Your art doesn't need to be pretty. It just needs to be something that'll help you remember, just to sort of relate what I've written down to an image. That way, if I ever have to bring it up during a test, I can remember the image and then hopefully remember what I wrote off to the right. So when we actually get to our experiments, we always start off with observations. And that's really what our entire first project is going to be, is making some observations. So hopefully you remember from our first class that an observation is something that you see, hear, smell, or feel. You can also taste it, but you generally shouldn't eat things in the lab. So one example that we might use of an observation is that the leaves are red, yellow, green, and brown in September. This is something that we can see. If we look at this picture right here, we can clearly see all of those different colors in the picture. So this observation should lead to your question. So observations lead to questions that design our experiment. So for example, an example of a question is, does a change in the amount of daylight affect the color of the leaves? So in your notes, you only need to write down this first part. If you would like to include the example, you can, but you don't have to. And I know in my notebook, I'm going to draw a question mark under what is your question, that just because that's really what this is talking about, is questions. So the example of the question here is, does a change in the amount of daylight affect the color of the leaves? This relates to our observations. We saw that the leaves changed, so now we're going to wonder, does the daylight cause that to happen? And then that question can lead us on to designing an experiment. So in order to design an experiment, you first have to write a hypothesis. A hypothesis is sort of a what-if statement. It describes what you think is likely to happen in an experiment. And in our class, we're going to write this in an if, then, because statement. So sometimes you'll hear that hypotheses are if-then statements, but we think it's important to add a because otherwise you're not making an educated guess, you're just making a guess. You should justify why you think it's going to happen based on the science you already know. So an example of a hypothesis using our example with the leaves changing colors, if the amount of daylight decreases, then the leaves will change colors because the chlorophyll breaks down when there is less light. So right now, don't worry about the chlorophyll part. You may not be familiar with chlorophyll and chloroplasts, but you should at least know the if-then statement. If the amount of daylight decreases, then the leaves will change color. This is our prediction. This is what our experiment is going to be set up. So now we need to talk about variables. Variables are sort of the bread and butter of experiments. These are all of the different parts of the experiment and everything that is going in to make your lab work. And these are often described as anything that changes in an experiment. So there are three main types. We have independent variables, we have dependent variables, and we have controlled variables. And this gets to our three upcoming questions, which is, what are each of these variables? So an independent variable is what you are changing or controlling in the experiment. It's usually the thing that you're interested in. So in our example with the trees, we were interested in whether or not the daylight was affecting the leaf color. So the daylight in this case would be our independent variable, or specifically the amount of daylight is our independent variable. A dependent variable is something that changes because of the independent variable. And it's usually what you're measuring or recording. So if you're putting down information about something into a table, that's usually going to be your dependent variable because it depends on what your independent variable is doing. So an example of this, again, back to our leaves, the colors that the leaves end up turning based on the daylight is our dependent variable. Another way that I like to remember the difference between the independent variable and the dependent variable is that the independent variable is what comes after the if in the if-then statement, and the dependent variable comes after the then in the hypothesis statement. So for instance, we have, if the amount of daylight decreases, then the leaves will change color. So that if, independent, sunlight, then leaves, dependent. They link together that way. Then our last type of variable are controlled variables. So in any given experiment, you probably only have one independent variable and one dependent variable. There are exceptions, but usually you only have one of each. Controlled variables, however, you should have as many of as possible. These are things that you are keeping the same between different groups. So for instance, if we have one group that's getting a lot of sunlight and another group that's not getting a lot of sunlight, we want everything else between those two to be the same so that we can be sure that the sunlight is what's causing the change. So for instance, if we use different kinds of trees, we couldn't be sure if it was the daylight causing the difference or the type of tree causing the difference. Or if we planted the trees in different kinds of soil, we couldn't be sure if the soil was doing anything to the leaves or if it was the sunlight. So in an experiment, you want to keep as many things the same across your different trials as possible. And those are all controlled variables. So I'll walk you through a couple of examples of how to identify the independent, dependent, and controlled variables in an experiment. So this example here says a student wants to study which type of cleaner kills the most bacteria. So the independent variable would be the cleaner that he is testing. So he's probably going to test different types of cleaner. Those cleaners are the independent variable because he's controlling which one he's using for any trial. The dependent variable is how many bacteria end up dying at the end, because he is using the different kinds of cleaners and then measuring how many bacteria die at the end. So the bacteria dead are the dependent variable. Some controlled variables that might exist for this experiment would be the type of surface that the cleaners are being used on, the number of bacteria that you start with, the amount of cleaner that you're using, or so on. There are many other different controlled variables that you could use because, again, you can have as many controlled variables as you want. Let's walk through another example. So we could also test to see if eating breakfast improves your test grades. So in this example, whether or not you eat breakfast is your independent variable because that's the thing you're changing. You'll probably take two groups of students. One group will have breakfast, the other group won't have breakfast. Then after that, you'll measure what their test scores are. That's the dependent variable because as a result of their breakfast, you want to see how they will score. One depends on the other, the dependent variable. Some controlled variables that you might have in this experiment, you might want to control the amount of breakfast that the breakfast eaters are having. You want to make sure they all have the same amount. You might want to make sure that they all have the exact same breakfast. You probably want to make sure that all of the test takers are taking the same test, they're taking the test at the same time, and that they're taking it in the same room. Again, you want as many things to be the same across the different trials so that only your independent variable could be changing the outcome. So finally, I want to talk about two different types of data, and this will be the last bit of notes that you take down. So we have qualitative data and quantitative data. Qualitative data tend to be observations, things that you see, hear, smell, etc., that can be written about, but don't have a number that can sort of relate to them. So for instance, if you're looking out and you are looking at the colors in a bag of candy, just listing the colors, you're seeing red, you're seeing yellow, you're seeing green, that's all qualitative data. However, if you were to count the number of pieces of candy in that bag, that would be quantitative data because now you're adding in numbers. Really, the difference between these two types of data is whether or not numbers are involved. Qualitative, no numbers, just words. Quantitative, must have numbers. Again, some examples. When there is less daylight, the leaves turn different colors like red, orange, and brown. This is an example of qualitative data. Because we're only talking about colors, there's no numbers involved. Another example, when there were 6 hours of daylight, there were 5 green leaves, 20 yellow leaves, and 16 red leaves. So even though we're still talking about colors here, this is now quantitative because we're also dealing with numbers. And finally, if we say that students who ate breakfast scored 20% higher on their math exam, that is also quantitative data because that 20% is a number, which tells us that we must have done some calculations with numbers to get there. It is quantitative. So this is the first 4 steps in experimental design. We talked about the question, we talked about making a hypothesis, we talked about an experiment. In many experiments, you'll also have a second step between the question and the hypothesis where you're doing some research. You're learning about some of the science that's already exists about your question. And then later, we'll talk about the analysis and the sharing of data. That will be the later part of our project that we will work on in a couple of weeks. So that is all. What you should do after this is go on Schoology and try the Ed Puzzle activity out. And when you're done with that, you should try the exit ticket, which is in the exit ticket folder. Please reach out to me if you have any questions, and I will be sure to try and help you out. Have a wonderful day, everybody.